I'm Daniel Levine, and this is the Bio Report.
The average kidney transplant recipient experiences kidney failure within 10 to 12 years after a transplant, putting them on a cycle that ends with kidney failure and a need for a new transplant. This cycle adds to strains on transplant recipients, payers, providers, and the healthcare system, and taxes the limited supply of organs for transplantation. Eledon Pharmaceuticals is seeking to extend the functional life of transplanted organs while reducing the side effects of current immunosuppressive treatments with its experimental first-in-class immunosuppressive therapy, Tegoprubart. We spoke to Steve Perrin, President and Chief Scientific Officer of Eledon, about the need for innovative approaches to immunosuppression in organ transplantation, how its experimental therapy works, and why it may also have benefits in autoimmune and neurodegenerative diseases as well. Steve, thanks for joining us.
Thank you, Daniel. Appreciate it.
We're going to talk about kidney transplantation, Eledon Pharmaceuticals, and the problem it's seeking to address with its experimental immunosuppression therapy. Let's start with kidney transplantation, though. How big a problem does a lack of available organs for transplantation represent today?
Well, the most well-known challenge of kidney transplantation is the severe shortage of donor organs. Despite thousands of lives being saved each year via kidney transplantation and an increase in the overall number of transplants per year, there's still about 100,000 people on the kidney transplant waiting list. And in the U.S. alone, approximately 5,000 patients die each year on the list waiting for a transplant. And that accounts for about one death for every 5 transplants that are performed. So it's a serious unmet need.
One of the challenges here is the life of an organ once transplanted is limited. How big a problem does that represent, and what role does a recipient's immune response play in limiting the life of a donated organ?
So despite advances in not only the surgical procedure itself, as we'll discuss in the interview here today, progress in immunosuppression, transplanted kidneys typically function for only 10 to 15 years. And that seems like a long time at face value, but if you think about it, most recipients are in their 50s or younger at the time of surgery, so many will require a second or maybe even a third transplant during their lifetime. And while they're waiting for another kidney, they end up back on dialysis, which costs about $100,000 per year. It's an overall cost of about $28 billion to the healthcare system to support patients on dialysis while they wait for kidneys. And they have a really poor quality of life for these patients. And in addition to that, a subsequent transplant brings increased complexity and risk. The organs usually don't perform as well. And it just creates it basically lops onto the overall shortage of organs that are available for new transplants.
There's been limited innovation around immunosuppression for organ transplantation. That may be a little surprising given how much effort there has been around immune modulatory therapies. Why haven't we seen a lot of innovation in this area?
So it's a great question, and I think to answer it, you really kind of have to take a historical approach because things have changed over the last 30 to 40 years. The unmet medical need was prevention 30 years ago of acute rejection, and to the extent of survival of organs back then, barely lasted 6 months, where acute early cellular rejection caused by the recipient's immune system attacking the transplanted organ caused a cascade of subsequent immune system responses resulting in kidney damage and subsequent rejection. So with today's standard of care medicines to prevent transplant rejection, the calcineurin inhibitor tacrolimus in particular, which was approved about 30 years ago, 1 year graft survival was the objective. It was even the holy grail because organs really weren't lasting out to a year. And initially, even though acute cellular rejection on tacrolimus was about 30% in the first trials with TAC, over the last 30 years, with optimization of tacrolimus levels in combination with other immunosuppressive drugs, acute rejection today is only in the high single-digit percentages. Acute rejections today are treatable with either a dose of steroids or a dose of thymoglobulin that translate deplete immune cells that are attacking the graft. However, long-term survival still remains at 10 to 15 years due to several factors, including nephrotoxicity of tacrolimus. So the same drug that is trying to protect the organ from rejection actually causes chronic long-term damage, resulting in organ rejection. And in addition, tacrolimus has several impacts on patients' health and quality of life, not only due to nephrotoxicity of the kidney, but it causes hypertension leading to cardiovascular disease. It's toxic to beta cells in the pancreas. It causes up to 30% of patients on tacrolimus to become diabetic. And tacrolimus has a few neurological toxicities as well that results in headaches, brain fog, and debilitating tremors. So that asks the question, what is the bogey today? What are we trying to solve for? Well, it's long-term survival past 15 to 20 years. Or again, the new bogey of the holy grail would be one transplant for life. And this is the challenge that Eledon is working to solve. And we're seeking to develop our drug Tegoprubart to replace tacrolimus as the cornerstone immunosuppressant by demonstrating superior efficacy as measured by graft function and graft survival, and superiority as far as safety is concerned compared to the toxicities that I just described for tacrolimus.
And what's the case for doing that? What's the case for developing improved approaches to immunosuppression as a way to extend the life of transplanted organs and help address this imbalance between supply and demand?
Well, despite the limitations of current standard of care immunosuppressant therapy, patients do much better after a kidney transplant than they do on dialysis, with its heavy burden on the quality of life. And the average survival on dialysis is only about 5 years. So it's quite an improvement over leaving patients on dialysis as an example. One promising avenue of research with the potential to extend the life of transplanted organs involves targeting new pathways. And one new pathway is the CD40 ligand pathway, which has been shown to play a central role in immune system activation and controlling inflammation in the body. Specifically, CD40 ligand is involved in the signaling pathways that regulate how immune cells, particularly T cells and B cells, communicate and respond to foreign antigens, such as from a transplanted organ. And targeting the CD40 pathway may provide significant advantages over current treatment options, including the ability to more precisely control immune responses and increase transplant durability with fewer side effects. By increasing this durability and lifespan of a transplanted organ, it should free up more organs for other people that are in need on the transplant list. And the longer we can allow organs to survive, fewer organs will be needed for repeat transplants and more organs will be available for people on that list.
Walk us through the process of immunosuppression. How are patients who are receiving a transplanted kidney prepared for that? And what's the long-term protocol?
Yeah, so as you can imagine, it's a fairly complicated process. The mainstay approach to the prevention of organ rejection is polypharmacy. It requires multiple immunosuppressive drugs combined together in a very specific fashion. And it starts off with what we call induction therapy around the time of transplant with high doses of steroids and a T cell depleting agent called antithymoglobulin, and then followed by long-term what we call maintenance therapy, where patients are on these drugs for life, and that usually includes a combination of low-dose steroids, mycophenolate, and the cornerstone drug that prevents rejection, which is the calcineurin inhibitor tacrolimus. However, before even undergoing the transplant, patients go through a very rigorous evaluation process that includes education about how the transplant's going to work, various tests that they'll have to undergo, discussions with clinical and surgical teams, including nephrologists. And the primary goal of the evaluation process is to ensure that each patient is healthy enough to actually receive an organ and is prepared to care for that new kidney over time. Assessment involves detailed review of a patient's medical and surgical history, along with tests to evaluate the function of vital organs, such as the cardiovascular system and respiratory system. Addition, there's compatibility tests, including blood tests, tissue typing, cross-matching to identify what a suitable kidney match might look like. And then finally, following the kidney transplant, the long-term protocol is for patients to take these immunosuppressive drugs and other medications for the rest of their lives to keep their bodies from rejecting the new organ. Patients undergo routine monitoring and doctor's visits to make sure their transplanted kidney is working effectively the way it should, and there are no signs of organ rejection, and they adjust their medications as needed for these optimal results. So kidney transplant recipients are expected to follow healthy lifestyles, including a healthy diet, exercise, to lessen the chances of comorbidities such as heart disease, high blood pressure, and high cholesterol.
And if I'm a physician treating one of these patients, how big an arsenal of immunosuppressant agents do I have to choose from, and to what extent are they tailored to the specific patient?
So I would argue that there isn't what I might call a personalized medicine approach here to the prevention of transplant rejection at this time. The primary arsenal, as I described, is this polypharmacy approach with multiple drugs, with tacrolimus being the cornerstone of the drugs that are typically used. There's guidance on how to dose each one of the 4 drugs that I mentioned, but each clinical site will play with the dosing depending upon the characteristics of not only the recipient receiving the organ, but also what the donor kidney looks like. There are a few additional drugs that are used in cases of problematic toxicity that are associated with tacrolimus, such as the nephrotoxicity. When patients just can't tolerate it anymore, they can be switched to a different drug called belatacept, which is a biological drug that inhibits T cell and B cell function through a slightly different pathway than Tegoprubart does.
How effective are the current agents that are used, and how targeted are they? I mean, when you're tuning down someone's immune system, does it have a systemic effect, and does that put them at risk for infections?
So the goal here is, as discussed previously, to push graft survival out beyond a year, and in the field, we've accomplished that goal. The new goal on the horizon is more of a long-term function and survival of organs past 10 to 15 years, and as I mentioned, the holy grail would be one transplant for life. Immune suppression is a significant concern in organ transplantation, presenting a delicate balance between preventing the organ from rejecting without increasing the risk of infections. Immunosuppressive drugs are necessary to prevent the recipient's immune system from attacking and rejecting the transplanted organ. So, therefore, careful management of the immunosuppression, including personalized regimens, because people metabolize drugs at different rates. They do this by close monitoring and preventive measures. And it's essential to optimize the transplant outcomes and minimize complications for each individual patient. Our approach with Tegoprubart is more targeted than standard of care immunosuppressants like tacrolimus that aim to wipe out the entire immune system or white blood cell population. Tegoprubart is designed to block CD40 ligand, a co-stimulatory pathway or molecule, which researchers have shown plays a central role in controlling how the immune cells that ultimately cause transplant rejection communicate with one another. And our preliminary data with Tegoprubart in patients undergoing a kidney transplant has demonstrated the potential for better efficacy as measured by kidney function, improved safety, and including reduced risks of the hypertension, diabetes, nephrotoxicity, tremors, and the other side effects that I described that are often associated with tacrolimus treatment.
And at a biological level, what happens in the body when a organ is transplanted? What happens to the immune system?
So there's a couple of different processes involved here that are different. So I'll describe each one separately and kind of talk about the highlights, if you will. So rejection occurs when a recipient's immune system identifies that the transplanted organ is a foreign tissue, which triggers an immune response against the transplanted organ. In the first 6 months post-transplant, rejection is often referred to as acute cellular-mediated rejection, which can be treated easily with either steroids or thymoglobulin. And it basically details the immune system from recognizing the organ, and the immune system can rebalance itself, if you will. The major cause of organ loss after 1 year is a different process called antibody-mediated rejection, which is driven by a different cell type, the recipient's B cells, which make antibodies that recognize the foreign organ, resulting in activation of several different biological pathways that then attack the transplanted organ, causing rejection and ultimately graft loss. Tacrolimus is not effective in preventing antibody-mediated rejection, so physicians attempt to remove the antibodies with crude tools such as plasmapheresis that basically filters your blood and removes protein from the blood to try to reduce the antibodies that are targeting the organ.
Eledon's lead experimental therapy is Tegoprubart. What is this and how does it work?
So Tegoprubart is an antibody that is designed to block CD40 ligand, which is the key receptor on activated T cells that causes that communication between activated T cells and B cells. It's the only anti-CD40 ligand inhibitor that's currently in clinical trials for patients undergoing both solid organ as well as cellular transplant, and including kidney transplant. CD40 ligand is that critical immune system protein that plays a central role in the activation that coordinates immune responses. And specifically, CD40 ligand is involved in signaling pathways that regulate how those immune cells, T cells and B cells, communicate and how they would respond to foreign antigens. In the context of transplantation, this signaling is a key driver of the immune system's ability to recognize a transplanted organ as a foreign organ that might mount an attack against it. A unique property of CD40 ligand is it has the potential to reprogram your immune system, if you will, so that the recipient's immune system no longer recognizes the organ as foreign, a process that's called tolerization. It's not entirely clear how tolerization works. However, we do know that blocking CD40 ligand not only prevents the activation of T cells against the transplanted organ, it actually converts those T cells to a different type of T cell called a regulatory T cell, which is a specialized subpopulation of T cells that actually suppress the immune system against foreign antigens and creates that tolerization that I was mentioning. As such, our approach is more targeted than current standard of care immunosuppression that really just wipes out the population of white blood cells and has demonstrated the potential for better efficacy and improved survival compared to current standard of care with tacrolimus.
And what's known about its efficacy from studies you've conducted to date?
So last week we presented an updated set of data at the WTC conference from our phase 1b clinical trial that's evaluating Tegoprubart in kidney transplant recipients. The results demonstrated that kidney function is measured by estimated glomerular filtration rate, or EGFR, stabilized after the first month post-transplant, and patients who remained on Tegoprubart for a year had a mean EGFR of 68. For comparison, the mean EGFR or kidney function for standard of care immunosuppression such as TAC therapy is about 53. So it's about a 20-point delta at the first year post-transplant. We also presented what's called preliminary iBox data. iBox is a composite biomarker of kidney function and predicts long-term graft survival with 1-year data. It's based on immunological responses and kidney function and a few other parameters associated with the kidney biopsy, and showed that Tegoprubart had a superior iBox than that's been reported for current standard of care on tacrolimus. We also... Yep.
I'm sorry, go ahead.
I'm sorry, Daniel. Go ahead if you'd like.
No, continue.
So we also presented, there was several other presentations at the World Transplant Congress with preclinical data that we presented in liver allotransplant, demonstrating that we markedly prolonged graft survival in non-human primates that were administered Tegoprubart as a key component of their immunosuppressive regimen. All the liver transplant remained healthy during therapeutic exposures to Tegoprubart with median survival of 587 days. About 25% of the primates survived long after drug withdrawal, including one animal that survived more than 2 years off treatment, again suggesting that Tegoprubart has the ability to induce transplant tolerance. In addition, blocking CD40 ligand has been shown in multiple different non-human primate xenotransplant studies to be essential for achieving durable xenograft survival as well.
And how targeted is this therapy? What's known about its safety?
So Tegoprubart is considered to be a targeted therapy due to its specific mechanism of action, blocking the activation of the CD40 ligand pathway has been shown to slow down disease progression and pathology in diverse preclinical models of both autoimmunity, such as rheumatoid arthritis and multiple sclerosis, as well as models of neurodegeneration. And the prevention of acute and long-term rejection by blocking CD40 ligand has been demonstrated in multiple species. And even today, after 20 years of studies using drugs that block CD40 ligand, it's the most effective way to prevent rejection in multiple species. So based on the updated phase 1b clinical data we presented at WTC, Tegoprubart continues to be generally safe, well-tolerated in patients who've received a kidney transplant. There's been no cases of death, no graft loss, no drug-related tremors, no new onset diabetes, which are the side effects that are typically associated with tacrolimus.
Since this is designed to target a receptor on activated T cells as opposed to the organ itself, how broadly applicable might it be to protect other transplanted organs and cells?
So significant prior research of tegoprubart and the inhibition of CD40 ligand as a whole has shown it may play an important role in immunosuppression in patients undergoing kidney transplantation and other types of allotransplantation or transplants from within the same species, human to human. We've also shown that it's very effective in transplanting organs from one species to another. So in the context of xenotransplantation, of utilizing pig organs as a possible source of organs to reduce the transplant list, has been shown to be quite effective there as well. As another example, we've shown that in the prevention of islet cell transplant in subjects with type 1 diabetes at an investigator-initiated trial that's being done at the University of Chicago's Medicine for Transplant Institute. In October, a positive initial data was reported from the study for the first 3 islet transplant recipients treated with tegoprubart as part of the immunosuppressive regimen. They demonstrated potentially the first in human cases of insulin independence achieved using an anti-CD40 ligand immunosuppressive therapy without the use of TAC or any other part of the standard of care, and really provided, if you will, a tentative cure for their type 1 diabetes. Among the 3 trial participants who received the transplant at the time of the data presentation, 2 had been functionally cured of their diabetes, achieving insulin independence and achieving normal hemoglobin A1C levels, a measure of average blood glucose post-transplant. And the third patient, who had only recently received his islet cell transplant at the time of the announcement, had reduced their insulin use by more than 60% and had shown good glucose control within 3 days following the transplant. Tegoprubart has been used as a key component of the immunosuppression therapy regimen in the first 2 patients who received a transplanted kidney from a genetically modified pig that occurred at Mass General Hospital in the last year. Tegoprubart is a key component of the immunosuppressive regimen supporting the longest survival pig kidney in a human. That patient is now out past 7 months. They've been out of the hospital. They recently threw a pitch at a Red Sox game. So quite amazing. If you had asked me a couple of years ago if a human would be walking around with a kidney 6 months, a pig kidney 6 months post-transplant, I probably would have laughed. Even more remarkably, we've been working with a research team at the Children's Hospital in Los Angeles who are currently using tegoprubart as part of the immunosuppressive regimen in preclinical studies in monkeys, evaluating the potential of cardiac xenotransplant in infants. Groundbreaking new preclinical data presented in April of this year show for the first time that xenotransplants of genetically engineered pig hearts may be a potential solution for babies born with fetal heart diseases and serve as a bridge solution while they're on the human transplant waiting list, waiting for an appropriately sized organ. And one of these animals in the study is out past 2 years post-transplant and doing very well.
And from a regulatory point of view, as you think about these other indications, do you need to seek approval for each organ you would use this drug in?
Yeah, that's a great question, Daniel. I mean, Eledon would need to seek regulatory approval for tegoprubart for each individual type of organ or cellular transplant. We're currently conducting 3 clinical trials in parallel of tegoprubart for the prevention of organ rejection in patients receiving kidney transplants. One is the phase 1b open label trial that I described the data that we presented a couple of weeks ago at the WTC. We also have a phase 2 trial called the BESTOW trial, which is a head-to-head superiority study comparing tegoprubart with tacrolimus. Patients were randomized 1-to-1 in that study, and the last patient enrolled last September. The goal here is to not only look at the superiority of kidney function, compare tacrolimus to tegoprubart, but to also look at the long-term safety profiles. And these patients have the ability to roll over into a long-term extension study. We're also supplying tegoprubart from an investigator-led trial at UChicago that I just described, evaluating pancreatic islet cell transplantations in patients with type 1 diabetes and for prevention of xenotransplant rejection around the world with multiple different collaborators.
There's a lot of interest in modulating the immune system beyond protecting transplanted organs. I know you're looking at tegoprubart as a potential treatment for ALS. How broadly applicable might it be in treating autoimmune diseases and diseases involving neuroinflammation?
So CD40 ligand is not a new target. It was actually discovered back in the late 1980s as a key biological receptor on activated T cells that, when inhibited in rodent models of autoimmune diseases, could prevent progression of those diseases and actually ameliorate their symptoms as well. That was shown in multiple different autoimmune models, everything from multiple sclerosis, rheumatoid arthritis, psoriasis, lupus, and many other autoimmune models in rodents in particular. These same mechanisms of CD40 ligand activation on T cells were discovered to result in activation of the peripheral immune system in animal models of ALS and Alzheimer's disease that ultimately results in neurodegeneration. Scientists at the ALS Therapy Development Institute showed that if you block CD40 ligand signaling with an antibody, you could actually slow down the progression of disease in these animal models, hence slowing down the progression of ALS. These data are what spearheaded the development of the second generation anti-CD40 ligand antibody that was originally called AT-1501 and that we now call Tegoprubart. While we have shifted our focus to organ transplant, we're still very committed to advancing the drug for ALS and are planning another trial once we've secured enough funding. And the vision here is to run a fairly large ALS study that could help us really know whether the drug is helping to slow down disease in patients with ALS or not.
Well, how do you think about prioritizing other indications? And is Eledon essentially a pipeline and a product company, or are there other candidates you're exploring?
So we're primarily focused on advancing tegoprubart in organ transplant rejection, given the significant unmet need here and the lack of innovation in immunosuppressive therapy for more than 30 years. The second rationale for the focus, which I described previously, is blocking CD40 ligand has been the most potent way to prevent both acute rejection as well as that long-term antibody-mediated rejection in multiple species, including mice, rats, baboons, rhesus macaques, and cynos. It's been a very prolific drug in multiple different species to prevent rejection, and now we're trying to replicate those goals as we translate tegoprubart into human studies. Our ultimate goal is to be the next cornerstone in immunosuppressant therapy and transplantation, regardless of the transplant type, whether it's a kidney, a heart, a lung, or islet cells, and regardless of the source of the organ, whether it's a deceased donor, a living donor, stem cell derived, or even a xenograft. We think that tegoprubart is going to be better and superior to current standard of care, tacrolimus.
Eledon's a public company. You had about $125 million in cash at the end of the first quarter. How far will current funding take you, and what's the plan for raising additional capital?
Yeah, so our current cash runway is projected to fund operations to the end of 2026. We're focused on achieving upcoming milestones, which include the top-line data readout from the phase 2 BESTOW study in November. We're also hoping for an interim clinical data readout from the investigator-led islet cell transplant clinical trial at University of Chicago Medicine sometime by the end of the year. And we're proud of the significant progress we've made as we enter the second half of the year and continue to achieve each of the key milestones we have guided to for tegoprubart in several organ transplant patient populations, including kidney transplant, xenotransplantation, and islet cell transplantation for patients with type 1 diabetes, as well as liver allotransplantation. And we're excited on what's coming up next and our anticipated key milestones in the next 12 months.
Steve Perrin, President and Chief Scientific Officer of Eledon Pharmaceuticals. Steve, thanks so much for your time today.
Thank you, Daniel. Appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.